हेलो। हेलो। नूजा मैम, आपको आवाज आ रही है मेरी?
Yes, sir. Am I audible?
यस मैम।
Okay, sir.
हरवंश सर। हेलो।
Good evening to all the participants, chief patrons, and course directors. I warmly welcome you all to the 1 week international training program, Plan 2025. My name is Anuja, and I have the privilege of anchoring today's valuable event. Many participants have joined us through our YouTube channel as well. Now, on behalf of Agriverse Green, I welcome our esteemed knowledge partners, ICR Atari Ludhiana and ICR Atari Kolkata, and our valuable collaborators, Chandigarh University, Biocar Seats, Akal Degree College, and Shran Welfare Foundation Trust. It gives me immense pleasure to welcome you all to the International Training Program Plan 2025, which provides practical learning in agriculture and elite next gen techniques being held from 18th to 24th December 2025. Plan 2025 is not just a training program, it is a movement towards transformative agriculture. Over the next week, participants will immerse themselves in cutting edge modules on digital agriculture tools, entrepreneurship, business, sustainable farming practices, integrated horticulture systems, climate resilience strategies, global bloom initiative, and inclusive growth modules and academic excellence sessions. Our vision is to lead a transformative movement in agriculture and elite sciences through ethical research, innovative technologies, and sustainable entrepreneurship that uplifts communities and ensure global food and resource security. Our mission is to empower researchers, entrepreneurs, and farmers by fostering academic integrity, advancing agri-tech solutions, and promoting sustainable practices in seed production, livestock, exotic crops, and resource management, driving inclusive growth and international competitiveness. The key objectives of this initiative is to provide participants with digital agriculture tools, including drones, AI, IoT, and mobile applications to enhance precision farming agribusiness by guiding participants agri enterprises, promoting sustainable soil health, and support long-term ecological balance, integrating horticulture systems across vegetables, fruits, and floriculture to diversify farm income and enhance agri ecosystem resilience, encouraging innovation and problem solving through hands on learning, case studies, and exposure to emerge agriculture technology, enhancing academic writing and publishing skills to empower participants to contribute to educational discourse and circulate impactful findings. With this, we built a collaborative learning environment that connects farmers, researchers, entrepreneurs, and policymakers for holistic agriculture development. With this, I now welcome our Professor Herban Singh Bariana. It is my honor to introduce Sir, who is an internationally acclaimed plant pathologist and one of the world's foremost authority on wheat rust resistance. Born in Nangal Ishar, Hoshiarpur, Punjab, Professor Bariana built the foundation of his distinguished scientific journey at the Punjab Agricultural University, Ludhiana, where he completed his BSc Agriculture with honors in crop protection and MSc in plant pathology. He later earned his PhD in agriculture genetics from the prestigious University of Sydney, Australia. Professor Brianna has served in leading scientific institutions including CSIRO, Agriculture Victoria, and the University of Sydney, where he dedicated 27 years to pioneering research on breeding wheat for rust resistance. He currently contributes to global agricultural science as an adjunct professor at Western Sydney University. A globally respected researcher, Professor Brianna has discovered more than 20 new rust resistance genes in wheat and developed closely linked DNA markers that have transformed marker assisted selection in breeding programs worldwide. His collaborative work on molecular cloning of rust resistance genes has been published in top tier nature journals, reflecting the depth and impact of his scientific contributions. He has trained and mentored numerous students and scientists from across the world, many of whom now lead major research programs in India and internationally. As a key member of the Australian Cereal Rust Control Program, he contributed to the team's recognition with the Barlog Global Rust Initiative Gene Stewardship Award. Professor Brianna has also co-led major Indo-Australian collaborative project funded by ACIR and ICAR, focusing on faster breeding technologies and broadening the genetic base for stem rust resistance against UG99. These initiatives have significantly strengthened wheat improvement efforts and enhanced food security for both nations. With his unparalleled expertise, decades of impactful research, and deep commitment to global capacity building, Professor Harbans Bariana stands as an eminent speaker for our training program, offering profound insights into sustainable disease control as a cornerstone of sustainable food production. With this, now I hand over the dais to Professor Harbans Bariana to begin with the 7-day international training program.
हेलो। हरवंश सर, आपको आवाज आ रही है? हेलो।
Uh-huh. Can you hear me now?
यस सर, यस सर।
Okay, that's fine then. I'll cut your phone. Yeah, good afternoon, everybody. And I will just take you a journey through rust control, which I have worked with many countries, but I was based in Sydney University for this work. Majority of the work was done at Sydney University, but my current affiliation, as Anuj just said, um, with Western Sydney University in adjunct capacity. So when it comes to plant diseases, there's a one big name called Professor E.C. Stackman. And he was the one who actually showed us that there are different physiological races or pathological races which exist in rust pathogens. And we need to know, because they are our enemy, we have to understand the enemy and then work against that enemy. So what he said was, in one of the publications, plant disease epidemics, outbreaks of insect pests, and bad weather are the greatest obstacle to assured food supplies for the people of the world. I think if we add another thing to that, that we have very short memories and we forget what has happened and try to be complacent once we get something controlled for a short while. So you can see these two photographs. This one is William Farrer, who was the Australian wheat breeder, I would say the pioneer, the first one. And he's on the $2 note, which is out of circulation now, but I had a copy of it. And he was a surveyor, but he came to Australia for a good weather because he has tuberculosis. So then he started his interest in breeding wheat for rust resistance. And his interests go back to working with Indian scientists. And if we look at his varieties, we can see pedigrees of some Indian, we can see in the pedigrees names of the Indian varieties, NP series and Calcutta series. And on the other side, this is Dr. Rajaram, who was also alumnus of University of Sydney. And unfortunately, he passed away during the COVID time. And he was the World Food Prize winner in 2014. And he worked all his working life at CIMMYT and probably the later part, which was shorter while in Ekada, which is Syria at that time. So these both are pretty much the top two wheat breeders who I can pay salute to. Oops. Slides are not moving somehow. Oh, yes, good. So now when we talk about rust diseases of wheat, we have three. Its first one you can see here is called stem rust because it comes on stems and it's caused by Puccinia graminis forma species triticide. And the second one you see around brownish, and this one is also called black rust. And these are the round spores. It's called leaf rust because it's majority of the time it makes the leaves full of it, but it can come on the rachis and other places to some extent. And third one, as the name indicates, and it's called brown rust as well. And as the name indicates of this, it's called stripe rust because you can see the stripes going along the veins. And it's called yellow rust as well because of the color in some parts of the world. And this is probably the most important rust disease of wheat at the moment globally. However, you never know. The others also come, but may not be noticed because it comes first and takes the varieties out and doesn't leave enough food for the leaf rust and stem rust. However, stem rust has been very, very damaging in the middle of last century when the epidemics happened worldwide. And especially, I can mention here the importance of stem rust in coming up in a national approach to rust control in Australia. So I'll just say broad outline of my talk is I will be talking about because the main, when we call it control or genetic control, I'm not touching here for the chemical control, but certainly in some circumstances, we can call it more like integrated pest disease management, in which chemicals do play a role, but our approach has been mainly genetical because chemicals were too expensive to be sprayed by all farmers. So the three components which I will be mentioning here is discovery of resistance genes. And of course, that will come with the identification of, with the availability of, rust pathotypes or rust races, which we call it. And then characterization of those sources, what do are genetically, how many genes control it, how pathological characterization work for them, can we do develop markers, DNA markers for those? And then finally, you have to deploy them in the new wheat cultivars. So the first two components will have no impact if we cannot deploy those resistance genes in new cultivars. And it equals breeding for disease resistance. And I must say, breeding for rust resistance or disease resistance for anything, any disease, it's a continuous journey. It's not a destination. We may think that we have reached the destination, but the new races, new pathotypes, they keep us uh, on toes to compete with them. So here you see one of my long-term associates, Gary Standen. He's doing crosses in the field. I thought I must acknowledge this because he worked almost 20 years with me. Now, if we look at how we can achieve those three things, discovery, characterization, and deployment, the main feature there is we have to have our handle on host research, which we have to find new genes. So they diverse genetically diverse genes so that we can put them in new varieties. And at the same time, we have to have pathogen research hand in hand, which let us identify new pathological variation or virulences in the rust races where the pathogen always try to go ahead of us in defeating the genes which we put in new varieties. And that is more like a game which is played between the host and the pathogen to sort of not look after each other, but to compete with each other. And it's our program, Australian Serial Rust Control Program at Sydney University had this model. But I think if we look at in India, there's a lab in Shimla and there's lab in Wellington near Ooty. They are doing the lot of pathological work for the whole nation. And then host research is continuing in many leading universities or ICAR centers. Karnal is probably the good example. And then universities like PAU, HAU, PNAU, whatever we call it, they other universities all, but I will not name the whole lot for the sake of time. So, and more recently, maybe about 15 odd years ago, or molecular genetics actually became integral part of host research and pathogen research. So we cannot ignore it. Whenever we look at the host, we do molecular genetics for mapping the genes. Whenever we look at pathogen research, we do look at the pathotypic evolution and look at what sort of background these different races belong to. And this is where a lot of DNA work goes in there. Now, the best part is this is basically discovery. But to bring the discovery to deployment, you need to have a germplasm enhancement program where do actually find new genes, and then you need diverse or most virulent pathotype to screen the material for so that breeder can actually select for resistance, which they can actually provide new varieties with good rust resistance to the farmers. So eventually it goes right up to the growers. And it's a two-way process. Farmers tell us about their problems. And we all together, I said breeders and others, agronomists come there too, extension people come there too. And any discipline these days, you have to have everybody involved to make the proper good team to deliver tangible outcomes. We cannot work in isolation. So that's how things can start here, go upwards, and come back in a two-way process that we understanding from each other and helping each other. So that's the model which I could say you can call it discovery to deployment model, you can call it breeding for disease resistance model, you can say rust control model, sustainable control model. You can sustain only when you have the all these components covered. Even if there's one missing, it will be very hard for us to compete against the pathogen populations, which, as Norman Borlaug said, rust never sleeps. So they keep on evolving all the time. So if I talk a little bit about how we reached the model in Australia, this model, that in 19, and I said to you that I will talk about stem rust in Australia. In 1970s, early 1970s, there was a major epidemic of stem rust in southeastern Australia. And in those days, it was thought that this stem rust is not a disease of the region. It's a disease of the northern region. And at that time, there was Australian states were really working like individual colonies. And if a pathologist or professor of pathology have to go from one state to the other, you have to have proper permissions. And one of the founder, actually, Professor Irwin Watson, who used to actually not take the university car but private car, and then try to develop relationship with the farmers so that he doesn't get caught. So he started surveying the nation in a very clandestine manner. So when that epidemic happened, he had already done some work together with Walter Waterhouse, who was his mentor. And I would say that Irwin Watson and L.M. Joshi, Dr. L.M. Joshi who worked at IARI on Rust for a long time, they were almost contemporaries with Professor Stegman at Minnesota, USA. So one of them went into pathology direction, and Watson went into more genetics and breeding direction. So then what happened that when those people were actually saying that Northerns might have put a curse on us by giving a stem rust, a disease which doesn't happen to exist there, eventually after meetings, a common sense prevailed, and this model, which I have summarized in a sort of more logical manner, came into existence. And we can call it more like a national approach to control diseases. It can be done for any disease, not only for rust, because models are pretty much same. Now, when we are talking about resistance types, there used to be people called qualitative, quantitative, minor gene, major genes, field resistance. There's about maybe 10 odd different terminologies used. But then people actually thought if we call the major gene or the qualitative resistance, which you can actually have either once the resistance is fully effective, it's more like your army because you have the full muscle there and you have the full protection unless somebody, it's more like a country's security. So if people are killed, then that qualitative thing is lost. And then when you go, it's called all stage resistance. Now it's accepted terminology, but people may still call it seedling resistance or major gene resistance. Then we call adult plant resistance or minor gene resistance. And it's often quantitative. It's basically you are reducing the severity of disease from 100% to maybe 70 or 60. But you need more genes to put together to make it acceptable. So just like defense of any country, we need army and air force together to have durable resistance or durable protection of a country. So this is where my approach always have been to have qualitative and quantitative resistance both bred into new varieties which goes to farmers' fields. Because, for example, if a major gene or qualitative gene breaks down, at least we have some level of resistance effective, which cannot let it go to 100%, but may take it to 50, 60, something like that, which is still not too bad. And that's where you can probably bring the chemicals in that kind of situation. But we we actually we have also advised our breeders or have been working together with breeders as pre-breeders to put more than one major gene in there so that if the gene breaks down, one gene breaks down, we don't have the total loss. So that's what the overall philosophy we have adopted in the Australian Cereal Rust Control Program, in which uh, my I was more like in charge of germplasm enhancement and screening. However, because of the guidance of PhD students, we have to work on all three areas. So that we can train the next generation of scientists, not only in discovery, but in characterization and in deployment. So that's where I'll give you some examples of how we have achieved it and going through the process of where we find resistance sources and how we characterize them, then eventually uh, make sure they are deployed. So main thing is that if you have published papers by discovery and characterization and your gene doesn't go to the farmer's field or your work doesn't reach the farmer's field, I think it's not it's not the true impact. And I think there was a poet, I'm sorry for English speakers, but it's more like an Urdu. So Muhammad Iqbal, he wrote, he actually said it in a very poetic manner. जिस खेत से दहका को मैसर नहीं रोजी उस खेत की हर खोशाए गंदम को जला दो. So which means that if a field cannot give the poor people food, then we don't have to work in that. So this is how I see that we have to put discovery and characterization together for the deployment of sources, which is very, very important and impactful. And it's a teamwork. You have to appreciate each other's lead, and so vice versa. Now, where do we get materials? So we have local, for example, in India, you would have a lot of varieties which may not be as good for all the things, but they may have resistance to a disease. So you can choose those. Then there was an effort about 15, 20 years ago. It started making new synthetics, like the current hexaploid wheat is actually a combination of A, B, and D genome. And that happened naturally in nature. Whereas synthetic hexaploids, people crossed durums or tetraploids where A and B genome with the D genome donor or Agilostotii and produced they repeated the history of producing hexaploid wheats with lot more involvement of different uh, yes please Karman Alam. Do you want me to answer your question now or later? Anyway, so those land race collections which were collected by various people like Babylov and Watkins from UK, uh, And this is example from the Watkins collection. You can see it's about 32 known and about two unknown areas. It was collect. You can see the representation of Afghanistan, Colombia, Myanmar. Oh, I mean, majority of the wheat growing regions were covered, and you can see overall picture on the globe that it was such a great genetic variation. And in those days, Britishers were ruling quite a bit of the world. And Arthur Watkins wrote letters to the diplomats, and those diplomats were asked to send seed to UK. And they had, I think, originally they had a lot, maybe about 7,500 kind of mark. But eventually, after purification, there's about 830 plus hexaploid beads and about 400 tetraploids, which includes durums and the other sort of tetraploids. So that this is more like going back to the basics. So what happened that when I was appointed to my role in Sydney University in 1996, I thought that 70 or 80% of my work is actually helping breeders and doing not a hardcore science. And I may not be successful in competing with people who have a lot of resources and they may have a lot more genetic variation in their hand, which I may not have. So I will not be able to compete with them. So after talking to some of my mentors, I figured out that I will actually start working on this collection. And interestingly, we have named quite a few genes, which I will come later part of the talk. And now there are a lot more people working on this collection. And I think Rust was probably my group was the better, well ahead of the others. And now we published a paper in Nature last year in which there's other all traits which a sort of Chinese and UK collaboration worked on, and they actually sequenced the whole, quite a large number of these, if not whole, probably whole collection, and they were trying to actually get gene mining out of that for a lot of traits. I think 137 or something like that, if I remember correctly, because that paper had a lot more than Rust. So I may have forgotten because I have been almost retired from that work for 3 years or 4 years. So that's the beauty of these collections, when uh, we actually can capitalize on those. But again, at the same time, finding those and then getting them into the better backgrounds is another challenge which we have to face. Now, well, I think the main thing is that you have to have national and international network for screening whenever you are actually doing when you want to actually be ahead of the pathogen, because you have to know what's happening in other parts of the world, not only in your part of the world. Here you can see I am with three, probably. This is the leader now, Sivasamy, Dr. Menon, Dr. Brahma. So I visited them quite a few years ago, and then we developed that collaboration, which we have very successfully achieved quite a few things. For example, in Kenya, they have this nasty race UG99, but quite a bit of material which is resistant to that, we can make it susceptible at Wellington. So similarly, if you go to Ethiopia, it's got a lot of pathogen isolates which can actually kill durum varieties. So we can actually collaborate with those guys for durum breeding. And then in Australia, we have our own trials. Then these are my colleagues working in Pakistan under the international projects. So basically, the theme of this is that, like the dialogue of that border film, "Hum hi hum hai to kya hum hai, tum hi tum hai to kya tum ho." Similarly, it works like two resistance types. It works like having good screening team worldwide, where you can actually access each other's facilities and help each other to protect the crops, to make it sustainable, so that that sustainability can lead to our food security. So that's what where sort of the point which I have tried to elaborate in my sort of title. So that's a lot of work, a lot of understanding with each other, recognition of each other, and your ability to actually get the best out of what you want to actually deliver to your local breeding community and eventually to the farmers. So that's the gist of the thing. Once that's the base where we actually start discovering new genes. So but then we screened this Watkins collection, which you can see that we have this is 3 years stripe rust data. YR is for yellow rust. The name genes are called YR genes. So you can see that here it's fully resistant, which is it's 1 to 9 scale. And I think up to here we can have major effect. So it's more like MR. This is MR, MS, MSS, S, and VS. So, and on this end is RMR, MR, RMR, R, VR. So we actually found full variation among that population from highly resistant or very resistant to very susceptible. And but this area was more of major genes. And when we come in this area, we still have the adult plant resistance, which is more like Air Force, and this is the Army. So here we get the genes which give you full, even if present singly, they can give you full control. But once you come here, you need to have 2 or 3 genes to find good level of resistance, which is commercially acceptable. For example, in India, you folks have very strong that it has to be certain number, I think, which is less than 20 or something, and you have to meet those things to get combination of these genes. Even one gene can give you that, but certainly it would be better if you have more of these. So I'll come to the next point of characterization. This is for stem rust, which we did. You can see again, we have variation from resistant to very susceptible. Then we go to leaf rust. We had again variation from very resistant to very susceptible. So which really was more like was a very, very welcome news for us that we can actually find new sources of resistance here, because that many lines, if they are resistant, we will be able to do something pretty much extraordinary and impactful. Then what we did was you can see this is just the methodology, how we spray with light mineral oil and grow plants in these small pots. And then this is only susceptible lines there, which is not my screening, but I took it because you can see the color of rust on when you have it under the greenhouse conditions. And we have 130 odd rooms which we can screen material. So it's more like a very large scale factory at Sydney University. And we took only the material which was given us a score of 4 or less, and we screened in the greenhouse to find all stage resistance genes. Now, and rest we have to the scores of 5, 6, and 7 we took where we assumed will be more like adult plant resistance genes. So the next step comes with that how we proceed. So as I said earlier that we have to be breeders, we have to be pathologists, we have to be geneticists, we have to be molecular geneticists. We cannot be all in one, but we can actually have a team which can, or we can train the students in a way that they get to touch the every bit of these things. So once you have identified which is the pathological screening, now it's genetics coming into play. So you are crossing a resistant with a susceptible, and you produce eventually F3 population, F3 population, which is the minimum you can start screening to see the progeny tested as well. Some people do F2, which I don't agree. And eventually we have taken to those F6 stage where at F3, the ratios of genetic ratios of segregation, they are zygotic. Whereas in F6, your ratios are gametic, which means for one gene, F3 is 1 is to 2 is to 1, which means 3 resistant, 1 susceptible. And but in F6, it's 1 resistant, homozygous resistant, 1 homozygous susceptible, maybe a little bit of heterozygosity there, but not much. And so you can see your precision at F6 is much higher than here, because here you have a lot of heterozygosity, because 50% of the population will not be fixed for the trait gene. So if it's solstice resistance, you are doing greenhouse, can be a single gene. If it's more than single gene, you have to then isolate gene 1 and gene 2, or if gene 3, which is eventually, it's a process of finding a heterozygous plant from here and develop line from there and develop populations which are only segregating at a single locus. Adult plant resistance, we do it in the field. It's hardly monogenic. It's majority of the times have 2 to 3 genes, if not more. Then you do actually the whole genome genotyping and quantitative trait analysis, and eventually you isolate. You can say that I will give you an example in the next slides or so, that you can find the chromosomes, but to isolate them as single genes, minor genes or APR genes, you can call it, or QTL, you have to do fair bit of work, which actually takes some time and nice facilities for screening. That's why we rely on screening at more than one place where we can do that work confidently. So now we have actually used the virulent races, or the most appropriate races or isolates or pathotypes of the pathogen, to get to this stage that how many genes, diverse genes, or how many QTLs we have identified. So then the next step is more of doing the detailed characterization. So here discovery is really trying to get an overlink with characterization. So then those mapping populations, or double haploids is another thing, or recombinant inbred lines, F6 are called same. Double haploids are produced by crossing wheat with maize, and then maize chromosomes eliminate, and then they double those particular plants, and they produce populations, and it's called doubled haploid. And then disease data, screening data we need, which I explained earlier. Then we take different approaches. Resistant, we take bulks of resistant lines and bulks of susceptible lines, the tail ends of the trait. And then eventually we can do high throughput genotyping. Or the better thing which we have figured out is that it's better not to bulk it and take a little bit more than 10 or 15. If you take 20, 25, you can get better outcomes and you can be there quickly. It's called selective genotype. And then bulk segregant analysis is another one, which is I mentioned here that you have bulks from resistant and susceptible. Then on these, we can do high throughput genotyping. We send it to the companies or labs where people have SNP technologies these days. And or earlier it was microsatellites people were doing in-house. And then diversity erase technology came in. And now I think SNPs, single nucleotide polymorphism labs are much more advanced because they have 35,000 SNPs, which is breeder friendly kind of markers, which are chosen based on the trait associations, or they have even in thousands and thousands, which are different arrays. But depends on how much money we have. It's only feasible to do depending upon the money. Whatever you have to design your things in proper way. I have been capitalizing on working with people who have the labs, good facilities, and tell them that we can work jointly on them if you can give us some discount, but we can actually work together on publications. So we can actually design primers to make the normal sequence tagged site markers, which are probably gone now, but now this is the one CASP markers, complete uh, allele specific markers, we call it, comparative allele specific. So you actually find resistant ones at one end, susceptible one on the other end, or one type is there, and any heterozygous come in the middle. So that's what the aim is. This is very high throughput system. Once you have developed anything you can do in-house, because now the technology is much cheaper. And so it's basically creating the linkage maps and then eventually identify the closely linked marker associations. So that's what the whole process of molecular mapping or identifying the genomic regions that control resistance. It used to be done in a cytogenetic manner, which was very tedious, but now it's much more easier. And I think almost, I mean, even most poor companies, countries, they have labs which can actually do that work for them. And I think it's gone much more cheaper than when it was 15 years ago or 10 years ago. So different technologies which I mentioned, RFLPs, restriction fragment length, it was the first. And then they went to polymer AFLPs. I mentioned these and explained the last slide, but this is the one which actually have shown here, and only single nucleotide difference. And this is the most powerful technique at the moment, which people are using wherever the genome, there's enough information available. I think all leading crops now have gone to SNPs, and nobody works on. There was Rapids also, but it's actually, and they call GVS also. These ones are converted to GVS, genotype by sequencing. So these are the sort of technologies which have been used, but now I would say that these two are the most important ones people use. Its DART is more likely called genotype by sequencing. An advantage here is actually it's based on the sequence of your two parents. But here you can sometimes get spurious SNPs which may not be polymorphic, but you actually try to get them uh, sort of by chance, statistical error or whatever you can call it. And so that's why there's a bit of competition in genotype by sequencing using DART technology and SNPs. But I think majority of the people are using this these days because it's more commonly available than this, because there's only one or two companies do it, this ones. So what we have achieved by doing those sort of screening and then developing populations and then using molecular mapping. So we actually had gene identified by YR47, and we made the cross for leaf rust. And once we located it on chromosome 5b, we figured out that Canadians have already done that work. We tried to relate the line number and we actually took it back to Iran. And so, but then we proved that these two genes are linked because then we thought we should test it for stripe rust or yellow rust. And we figured out this gene was pretty closely linked. You select for this, you select that one also, you select for this, and you select that, which is dual resistance by selecting for one. And this one was intermediate effect, even though we can see it in the greenhouse. And these two genes were present in one line, in one genotype, and we have to isolate them to get two components. One we located on 4A, others on 3B. And you can see here by R57, it's actually flanked by this marker and this marker. This is more like SNP. This is a SNP and this is microsatellite. And both markers worked equally well when we validated. I'll come to the next slide. So then 63 is another gene which we have sort of reached. We took a different approach to map it. Initially mapped it, and then we took a different approach to find out what actually it is, which probably takes me full lecture to explain. Then we have YR72 again. Intermediate effect, you can say that we have 1, 2 intermediate. Then we found one, which is a adult plant resistance, and then 2 more intermediate effect genes. So you can see that from this collection, and there's a lot more in the collection, which, if I would have worked another 20 years, or some people take my work further, they can find out. And these are the sort of things where a lot of our students involved, and we trained them on all those aspects. We explained them the discovery, but when they were screening it, they understood how discovery can be made, but characterization was their major work. And then these genes brought into our germplasm enhancement, and you have seen Gary Standard doing crosses in the second slide. So here are some of the variations. Some genes actually pretty much, this is the susceptible control. Some genes look pretty close to susceptible, but in the field, they look much better. And but in the seedling, they don't express as well. But certainly, once you have worked your lifetime on these genes, you can actually differentiate this from this very easily. But certainly, there was one thing which I must explain. There was a gene which we worked on, not from this collection, but we called it YR34. And then we showed that it was associated with onless character in wheat. And this was in this range. And somebody from USA, they actually found the gene on same chromosome, but that was recombined with the onless character, and they said they have a different gene. But we figured out eventually by collaborating with each other, by making crosses, that those both genes were actually the same, but there was some kind of recombination. And rather more than that was that there was some rearrangements which is present in which we worked on comparative to the USA. So wheat is a very, very mixed plant because it was a hexaploid. It was derived from natural hybridization of a tetraploid and a diploid. And so a lot of genetic variation has actually gone in different ways, which people have captured in the, whatever way they have taken that approach. So now, just to give you an example of that, the sort of mixing up of the genomes. So this map is the 2D map once we compared. And this is the 2A map. And this is the 2B map from the standard comparative maps, which we sort of try to look at. And if you look at this map, which we created, it was for landrace with landrace. It's number I have put, but names were different. So you can see how it starts from here, and then goes all over. So now if somebody finds the same gene somewhere here, he or she may that it's new, but it could be the same when it's here. So uh, this sort of mess, when you compare the land races and the new varieties, you will come across. Now I talked about validation of markers. As I said, showed you this SSR marker and this SNP allele. You can see in resistant, this base pair was present and A allele was present. And once we tested on quite a few Australian and some Indian cultivars, we found that the marker works really, really nicely. And this work was done by one of the Indian students who worked with us as a John Allwright Fellow under the ACIR project, Mandeep Randhawa. So you can see that once you haven't seen the resistant allele in any of these, that means you can use it very, very effectively. And same here. So if you have SNP system working, do this one. If you have the older gel-based technologies, you can use this one. And then I will come to, I think it may be a couple of slides further. So just with an example that with minor genes or adult plant resistance genes, we can actually see these ones was in a variety. We actually figured out there are 3 genes. We cannot see the segregation properly because you need larger population. But when we did the QTL analysis, we found one gene on chromosome 1VL, short long arm, one on the short arm of 2A, third on the short arm of 3B. And this gene, once we looked at, it was giving some effect like nearly susceptible, but we can see bit of chlorosis. And we actually figured out that we actually related that phenotype to this kind of, this peak markers by comparing the phenotype of the seedling in the greenhouse and by comparing the genotype of this, segregation of this peak marker. So this is how you cannot get that it's on this chromosome, but once you have 3 genes, you will get these peaks like that, and then you see which contributes. So we know that in all 3 cases, only one parent contributed, but it could, if it would have been contributed by the second parent, this should go this way. So that's how this algorithm or program works. So then in this case, what you do is for the validation, when you take lines for the peak marker allele, getting all the 3 genes, 2 genes combinations and singly, you can see when you have 3 together, how good it is. Once you have 2 together, better variation, but it's high. 3 together, that year may not have been very good because it's low in everything. Then go there, it comes. But once they are singly, they go much higher. So that's what validates the work which we do. That's certainly what we have identified is correct. And it's a pretty much validation of the quantitatively inherited traits or APR genes, which we figure out by sort of comparing the genotyping, the peak markers, and selecting the lines based on that and looking at the phenotypic data. Then coming to the cloned ones, SR33 was cloned by one of our students, who is professor now in one of the universities in Australia, Dr. Sambasivam Perianan from TNAU. And when he was successful for a scholarship with me, I told him that after 1 year, you will go and work with the top molecular biologist who I was working closely with, Dr. Evans Laguda. So we cloned this gene, and it was NVSLRR for those who understand these systems. And then we used this gene as a bait, and through using different technologies, we cloned these two, and we figured out they are similar type. And then in another technology which was developed in collaboration with the John Innes Center in UK, we figured out another way of cloning called agrensic association genetics, and something sequencing. Rensic is the short word for that. And you create mutants, and you go through all those baits, and then this one would have been the first gene to be cloned, but this there was one recombinant which was not explainable, and this turned out to be much later. And this is adult plant resistance gene, which is actually giving resistance to leaf rust, stripe rust, and stem rust, and even powdery mildew. So it's a minor effect gene. It's not. It's more like 60% disease rather than 100. And by year 15, I worked with a Finnish and Israeli group, and that was the Israeli person working in Finland, and we cloned that, and we figured out it's a tandem kinase. And majority of the time, at the moment, our knowledge says that quite a few genes, which are maybe not many, but half, maybe 5 or 6 cloned, they are in the kinase families from the tetraploids. So that's the sort of work which we have done in collaboration. It's not that I am the molecular biologist who did it. No, I had the material, I had the thinking, and the people have technology and they had the thinking. So we shared that material and ended up publishing it together in high profile journals. So I could not have done it alone. Those guys could not have done it. And by year 15, even those guys lost the material, while we took it from Israel, but we still had the material, and then give them in different backgrounds as well. So deployment, conventional phenotype based, you can see here that once you have a gene like this, and this, you combine them, it gets so clean that this is the light actually. And then susceptible is here. So that was my PhD work. And then marker assisted gene pyramiding, this website, MAS Wheat, they actually put all the market details, and information is available to everybody for looking at using those markers, and all the protocols are given on that website. So now this is again work of Mandeep Randhawa, and you can see we actually try to combine these genes in these ones in these varieties. So we try to keep these genes which were present in the original variety, and we try to put these in combinations. And we actually have that work was not quite finished, but which we finished later on. We actually used a couple of Indian backgrounds also, because they are part of the ACIR project. And we later on delivered that material through Canal Group, Indian Institute of Wheat and Barley Research. I think at some stage it used to be called DWR. So these genes, what you can do is, this one is one kind, this one is giving you, hexose transporter, and this is the tandem kinase. So once you are putting gene cassettes together, you can put together different types on your cassette and transform but which is not acceptable to the community at the moment. But certainly people have combined up to 5 or 6 genes for stem rust, and these combination of certain genes and cassettes, and the concept is proven but it's not acceptable to the farmers, to the community, and but certainly, if the need be at one stage, that those protocols can be followed. And so now I think it's end of my time. Maybe I might have taken a couple of minutes more. And it's field, greenhouse, lab. All these three technologies we need. We cannot succeed with one, and we have to have things working, things achieved working together. So I think I will leave now some time for the questions, and thank you very much for listening and appreciate your time.
Thank you so much, sir, for such an insightful session. Now, this is a doubt clearing session. If you have any questions regarding today's topic, please drop your message in the chat box. I repeat, if you have any questions regarding today's topic, please drop your message in the chat box.
Couple of people had hands up. I have forgotten the names. Uh, so, Anuja, can you speak the question, or I have to look at in the? Ah, uh, thanks. Yeah, I think it's better if Anuja, you can tell me the question, because this is basically all thank you quest things, and I cannot see any question there. Ah, Shivam Kumar. Yes. I actually was offered a scholarship to work with Professor McIntosh at Sydney University. And even before that, at MSc, I did with Dr. Sokhi at PAU, who was student of Dr. L.M. Joshi. And uh, and then based on my Rust work, I was selected for that scholarship. And Dr. McIntosh was the world leading authority in Rust. And so basically, I never saw any issues, but certainly I have to put my head down to learn. A pathologist doing cytology and then making a way towards doing a bit of DNA work was a challenge, but certainly I took it and I did not see any major issues. Okay, now Swash V, how are CRISPR and genome editing technology transforming plant breeding? Yes, I have not included that in my talk. We have proved the concept of non-GM CRISPR gene editing. So what we did was that we actually used this paper is on the bioRxiv, and it needed some questions to be answered, but somehow our team basically went in different directions, and it's still sitting. One day somebody will take up the challenge to answer those and maybe publish it in another journal, but we put it on the bioRxiv. And we actually used uh, we, we understood the pollen biology, and we used RNP complexes and nanoparticle complexes through solution. We directly pollinated the plant, and which turns out to be it's not seen as a GM crop. And it's certainly a lot of challenge yet, but I think I have seen a lot of papers coming up these days on some other traits, and but haven't really seen anything in wheat which is routinely used. But certainly at the experimental level, it's there. Gene manipulation and GM cross effect is susceptible. Look, when we are actually using the changing, manipulating the sort of genome, things can happen because if you know, some people have published on phenotypic specificity, and they did domains popping, and so it's basically if the domains are deleted by the CRISPR-Cas, and it can certainly lead to bad things, susceptibility, but if it's done for the good purpose, they may lead to resistance as well. So it's an area which has to be explored. So why is mark-assisted pedigree breeding less explored? I think this answer is less explored on marker-assisted. Is that basically money makes the mayor go? People have, if you have a lot of money, you will spend it, and now it's working. And I could have included some slides where we have done that DNA isolation, where we used to we started with about $8, or I think $8 or $5 per plant per line, and it is now, 50 cents. You have high throughput systems available, and people are now using a lot of marker assisted selection in early generations so that they can actually enrich the population. They can shift the population balance toward the positive alleles, and then they have to go on a larger number of trials than larger areas on those, rather than a lot of numbers, but then not that many trials. Okay. Yes, Dr. Baniwal, you have a question? Who can see your messages recording on? Uh, I mean, everybody has seen it. So I don't know what exactly you meant, but I think people have people who have here. But if somebody else is interested, why not? But as long as proper, if they want any information, they should actually consult, because some of this information may yet be unpublished.
हेलो।
Yes,
हेलो।
please.
जी सर, राजेंद्र बेनीवाल सर आपसे बात करना चाह रहे हैं। थोड़ा उनको ना दिक्कत आ रही है
Hmm.
अनम्यूट करने में।
Jarur, no problems. Dr. Baniwal, how can I help Gurpreet and Dr. Baniwal here?
यस सर। मनप्रीत जी,
How
आप
can I
उनको
help?
अपनी तरफ से उधर से अनम्यूट करके देखिए प्लीज। मनप्रीत जी।
Oh, where is
जी हो
he
गया।
on the
जी
system?
डॉक्टर साहब।
Well, yes, Dr. Baniwal. Okay, I can't hear you. Yes, Dr. Baniwal. I can't hear you. Please speak up a bit. Looks like it's too cold there, eh?
सर आपकी आवाज नहीं आ रही बेनीवाल सर। सर आपकी आवाज नहीं आ रही उधर से।
So I believe we don't have any more questions and we're not able to connect with Benewal sir. So I'll begin with the word of thanks. Now I also warmly welcome and thank each one of you to this week of learning, collaboration, and innovation. May Plan 2025 sow the seeds of knowledge, nurture the spirit of entrepreneurship, and harvest the fruits of sustainable growth for generations to come. Now it's time to extend my special thanks to Professor Gurpreet Singh, CEO and founder of Agriverse Green, for taking the initiative to organize this event. I also express my sincere gratitude to Dr. Rajinder Beniwal, Dr. Gurshaminder Singh, Dr. Amandeep Kaur, Lavjeet Singh, Gurdeep Singh, Manjot Kaur, and Arshdeep Singh for their valuable support and contribution. Let's make the event successful by joining the hands together. Once again, thanks to all of you for being part of Plan 2025, and thank you for your patience, trust, and precious times to be with us today. Once again, thank you, everyone.